bacteria, the polymerase stops transcription at the end of terminator and the mRNA can be translated without further modification. But in eukaryotes, RNA polymerase II transcribes the polyadenylation signal sequence. The RNA transcript is released 10 to 35 nucleotides past this polyadenylation sequence. So in the eukaryotes, there is a signal called polyadenylation signal, and the RNA polymerase transcribes the polyadenylation signal sequence, and it basically goes beyond that, and the RNA transcript is released 10 to 35 nucleotides past this polyadenylation sequence. Okay, as we said, the processing also takes place in the eukaryotic transcription. There are three steps in the processing which takes place. One is the addition of the poly A tail, which takes place at the 3' end, and there is also addition of 5' cap on the 5' end. So mRNA has two ends, 5' end and the 3' end. So on the 3' end is addition of a poly A tail. So what's a poly A tail? It's basically the it's a stretch of A's end. What is A? It's adenine. Okay, so there is a on the 3' end there is addition of adenines, and it forms a kind of a tail. So it's called a poly A tail. And on the 5' end there is addition of a nucleotide which is a modified form of guanosine, which is added on the 5' end of the mRNA. So why are the two ends of mRNA modified? The two ends of mRNA are modified to make mRNA more stable. Why? Because mRNA is very sensitive molecule, and mRNA has to move from nucleus to the cytoplasm, and on the way there are many enzymes, and those enzymes can degrade mRNA. If mRNA is degraded, then no protein will be formed. So that is why, to make mRNA more stable, the two ends of the mRNA, they are modified. So this is basically the poly A tail. Okay, 50 to 20 adenine nucleotides added to the 3' end. So this is the 3' end. Here 50 to 250. So this is the 3' end. On this side 50 to 250 adenine nucleotides are added here. And this is the 5' side. A modified guanine nucleotide is added to the 5' end. And basically in the mRNA which is made, there are some regions called introns and there are some regions called exons. Introns are basically a kind of a junk DNA. Okay, so exons are the ones which codes for proteins. So introns, or you can remember introns from something which is interfering, it's also called junk DNA. So remember that we're talking about mRNA, but that information in mRNA is coming from DNA. So the part of the mRNA which is coding for an intron is coming, that information is basically coming from DNA. That information is coming from DNA. So there are introns and exons in DNA, and when they coded in mRNA, those exons, they are removed by a process of RNA splicing. We are going to talk about that, and that takes place with the help of a complex of RNA and proteins that's called spliceosome. Introns are removed and all the exons, they are joined together. Okay, so I think it's shown here. It's shown very nicely in this picture. So this is the your mRNA. Here 5' cap is attached. Here poly A tail is attached. You can see that this is light part, this is intron, this is intron, this is axon, this is axon, this is axon. Okay, all the dark parts you see, these are axons, and all these light parts you see, these are introns. So during splicing splicing, these introns, they're removed and all these axons, they're joined. So you can see this is all coding segment. Okay. So so this is the mature form of mRNA. Okay. This is not a mature form of mRNA. This is a mature form of mRNA because in this, there's a 5' cap, there's a poly A tail, and all introns, they have been removed, and the axons, they have been joined. They have been joined, and you can see when all the axons, they have been joined, they form the coding segment. And this is the mature form of mRNA. And also there's important thing to know in the mature form of mRNA is that on the 5' side, there's a 5' UTR, and on the 3' side, there is a 3' UTR. Now, what are UTRs? UTRs are untranslated regions. Okay. So, we don't know why mature form of mRNA they have. We don't know their function yet, but they may have some role in some kind of regulation. But they are there in the mature form of mRNA. So this is the structure of mature form of mRNA. As I said, as I talked about RNA splicing, which takes place with the help of spliceosome. What's a spliceosome? It's a complex which is made of RNA and proteins. So you can see this is a spliceosome. This is a spliceosome, which is a complex of it's a complex of proteins. So this is a protein, and this is a complex of RNA and proteins. Okay. So these are actually called sRNPs. These are small ribonucleoproteins. So this is a complex of RNA and protein. So these are all sRNPs, small ribonucleoproteins. So they are complex of RNA and proteins, and they are further complex with other proteins. So that forms a spliceosome. And the function of this spliceosome is to remove the introns, and it helps in the joining of the axons. So this is, you can say, intron is removed, and the axon is joined together. Okay, so basically, how does splicing takes place? It takes place with the help of these spliceosomes, and in these spliceosomes, there is a RNA, and this RNA acts as a catalyst. This RNA acts as an enzyme, and when RNA acts as an enzyme, they are called ribozymes. So ribosomes are catalytic RNA molecules that function as enzymes, and they can splice RNA. Now, these are some of the properties of RNA which enable it to function as enzyme. RNA can form a three-dimensional structure because of its ability to base pair with itself. Some bases in RNA contains functional groups that may participate in catalysis. RNA can may hydrogen bond with other nucleic acids. So these are some of the properties of RNA because of which RNA can act as a catalyst. Okay, there's another process which is called alternative splicing. Alternative splicing is, let me explain you from this picture. So basically, if this is a protein, if this is your mRNA, and it's shown here as a DNA or a gene because the information or whatever is there in the mRNA is actually coming back from the DNA. So if this is one exon, this is intron, axon 2, axon 3. So during the transcription, these introns will be removed and these axons will be joined. Now it means that in the mature form of mRNA, there will be axon 1, axon 2 and axon 3. Now axon 1, axon 2, axon 3, how they are arranged? It can be arranged axon 1, 2 and 3 together. That forms one kind of a protein. There's a possibility that after axon 1 comes the axon 3, then comes the axon 2. Then it will be like axon 1, axon 3 and axon 2. Okay. Then it will be like axon 1, axon 3, axon 2. It is going to be a different protein. Or maybe it can be axon 2, axon 3, axon 1. It's going to be different protein. So how these axons they are arranged. They form on the basis of the arrangement, they form different proteins. So this process is called alternative splicing. Okay, this process is called alternative splicing, which means how these axons are joined, it can be in a very alternative pattern, and because of that, different kinds of proteins can be made. And basically, every axon codes for a domain. Now, what is a domain? Domain is that part of the protein which can domain is that part of the protein, of a fully folded protein, which can perform an independent function. That is what a domain is. So domain is that part of a fully folded protein which has attained a three-dimensional structure, which can perform a independent function. Okay, that is what a domain is. And every domain is coded by a axon. So this is what is being shown here. Okay, the next part is translation, and I'm going to make a separate video for that.